Hi everybody, my name's Maddie and this is Bianca, and we're both students here at Colorado State University. We're both microbiology majors, so we've taken an immunology class, and one of the things that we thought was really hard was VD&J gene recombination. So what we're trying to do is help people that are coming along after us to try and understand this concept. So we're going to start off by making a model so that if you learn kinesthetically, you can try and sort this out on your own. By using something physical that you can put your hands on to try and get a better understanding of it. So right here is going to be the beginning of our model, and it's a chromosome. It's actually a pipe cleaner, so if you want to go out and make your own model, it's pretty inexpensive and easy to go do. So this is going to be the very beginning of our DNA. Okay, so as we continue on making a model, you can see that we still have our white long strand of DNA, but now we've added some different gene segments to each portion of the DNA. So these red segments up here are called the V segments, and that stands for variable segments. And then here, this green, we have the diversity segments, followed by the joining segments, and then these longer blue ones here are called the constant segments. Now you can see that there's a heavy chain and a light chain, and those correspond to the different portions of the antibody, or the B or T cell receptor that VD&J recombination is critical for. The main differences between the heavy chain and the light chain are that the light chain do not contain any of these diversity segments here. And you also will notice that there are joining segments in between every single constant segment, whereas up here on the heavy chain, all the joining segments are together, followed by all of the constant segments. So just to put this in perspective, you can see that here's the light chain on an antibody, and here's the heavy chain. The heavy chain extends all the way down, whereas the light chain is just this portion of it. However, on both the heavy and the light chain, you have this portion up here that's called the FAB region. And that FAB region is also known as the variable region. And that changes whenever VD&J change, and that makes it a lot more variable, and it's exactly what binds to the antigen. So as you can see, our model is getting a little bit more complex, but don't get confused. You still have your V regions, D regions, J regions, and then the constant regions down here. The only thing that I've added is an RSS sequence. RSS stands for recombination signal sequence, and what that does is it alerts the different gene sequences to each other so that they know where to come together during the VD&J recombination. In this model, the blue and the purple segments that you see here are showing the RSS. So RSS, there's two different types, as you can tell because we have two different colors, and that is based on their size. So the blue RSS sequences that we have here are 12 base pairs long, next to the variable region and next to the joining region. And these purple ones here that are on the two sides of the diversity segments are 23 base pairs long. The rule that you want to remember is that 12 wants to go to 23. So whenever these guys are coming together, the blue and the purple are going to want to go to each other. So logically, if you look at this, the V and the J aren't going to go together because they're both 12. The V is going to go to D, and then D is going to hook up with J, because that way blue and purple, purple and blue, 12 and 23, 23 to 12. That's the best way that they like to hook up. And just to keep it clear, remember that this is going to be a heavy chain because we have the diversity segments here. Okay, so hopefully now you understand how to make the models, and I hope that that will help you to use whatever style of learning you want in order to help understand this concept a little bit better. But before I start showing you the actual physical happening of the VD&J recombination, let's just make sure that we understand what exactly is happening. So the whole point of VD&J recombination is to add variability to B cell receptors, T cell receptors, and antibodies. And that's really important because each antibody, B cell receptor, and T cell receptor only binds to one antigen. So they all only have this many segments, so they all have to hook together in different ways so that they can be unique and bind only to that one antigen. And just to help you logically process this, this is going on for B cells, which are making the B cell receptors and the antibodies. That's going on in the bone marrow. And for T cells, it's happening in the thymus. So here we're seeing our light chain again. Remember, these red segments are the V, the yellow segments here are the J, and then these dark blue segments are the constant regions. This is the light chain with those RSS sequences on that we talked about before. So the purple, as you remember, are 23 base pairs long, and the blue are 12 base pairs long. And remember, the 23-12 rule means that they want to match up. So what happens during the actual VD and J recombination, in this case with the light chain, because there is no D segment, it's just going to be VJ recombination. So these segments, the blue ones, are going to want to match up with the purple one. So they match up here, and then what happens is that these two proteins here, which are known as RAG1 and RAG2, what they do is they help to line up the purple and the blue segments, and then they help to cut it. So what would happen is that this segment here, these purple and blue match up, and then you get a cut right across there. So just to demonstrate the different variability that you can have here, let's say theoretically that V segment 3 was going to be paired up with joining segment 4. So this joining segment, led by its RSS sequence, would line up with the V segment 3. They would match up there, and then this section of the DNA would be cut right here. This would be taken off into a loop, and this section here would be left to repair itself using another protein, which we'll talk about later. Or, hypothetically, in a different scenario, let's say that V segment 2 is going to be combined with joining segment 3. So in that case, you would get the RSS sequences lining up again. In this case, this snippet of DNA, this loop, would be cut off, and these two portions of DNA would be left to recombine themselves. This is one way of explaining how the variability happens, because clearly all of these different segments, and there are hundreds of them along the chromosome, they can all combine in different ways, and that's how you can get that extreme variability that allows for antibodies, B-cell receptors, and T-cell receptors to identify such unique antigens. So, as I said before, RAG1 and RAG2 are critical in this process. So let's say that V segment 2 is going to be working together with J segment 3. So in this case, our RSS sequences are going to line up. You can see that the blue and purple 12 to 23 rule, that's critical, are lined up there. So this is what RAG would be doing, but since my rags are made of pipe cleaners, I'm going to go ahead and use scissors. So it would cut neatly right there, getting rid of those RSS sequences. So now what we have is this DNA left over and these two chunks of DNA, which have now been cleaved from their RSS and are kind of hanging loose here. So this DNA here, which is not going to be used at all, is going to form a loop. And it kind of forms like a vector, a plasmid, and it just goes off into its own space and is not used. The more critical piece, however, is right here. So we've got this hairpin here, which you're just going to have to imagine, and the rags are here, and then this third protein comes in, and it's pretty critical. It's known as TDT, and that TDT stands for terminal deoxynucleotidyl transferase. And what happens is that this TDT works here in between these two, and what it does is it fills in nucleotides, and it ligates these two pieces together. So now you see that our V segment and J segment have been successfully joined together. We have these other constant regions down here, another V segment up here, and so that's how the V and J segments are joined together on the light chain. So here's the heavy chain, and I'm going to show you how that happens. This time we can actually have all V, D, and J combine. Remember, you have your red sequences here, the variable, or V, D for diversity, and the yellow here are the joining, and then down here these dark blue ones are your constant regions. So as you can see, again, just as a reminder, you have the RSS sequences in the light blue and the purple. The light blue is that 12 base pairs, the purple is the 23 base pairs. Those RSS sequences are critical for lining up the gene sequences so that they get combined correctly. In the heavy chain, the way that this works is that the diversity sequences, or D sequences, join up together with the J sequences first. So we're going to have our RSS sequences combine, and then as you remember from the light chain, you have that RAG1 and RAG2, which are critical in having these guys line up properly and helping them cut correctly. So RAG is technically cutting these, but I'm going to go ahead and do it with scissors. So this forms a loop, and that loop kind of just goes off on its own and is no longer of any consequence. So now we have our J sequence here in yellow and our D sequence right there in dark green. So that TDT, or terminal deoxynucleotidyltransferase, is an important protein here because it comes and it forms kind of a glue between these guys by putting in extra nucleotides and ligating those together. So now we have our D and J joined together, but it's critical, we got to get the V segments in there as well. So we can have our V segments come over here and have those RSS line up again. So we've got blue and purple because remember we're following that 12 to 23 rule. RAG1 and RAG2 stepping in again to help cut this out. So now this is going to form a loop again, just like the other one did, and he just goes off into the cytoplasm. So now we have these guys as they were joined together, and now our V segment is joined together here again by the terminal deoxynucleotidyltransferase, or TDT. That protein is what helps fill the gaps with extra nucleotides. So as you can see, you can get a lot of variation because any one of those red variable sequences could have joined with any diversity segment and could have joined to any joining segment. Almost any combination can happen, and that's how you get all of the variability up there in the top of your antibody, your B cell receptor, or your T cell receptor.